Okay, so what about the immune response? So we know, obviously, the immune response is very important. And one of the things that we're involved in, these are studies being led by Paul DeBacher at the Broad Institute, and these are sort of genome-wide association studies. So this is looking at the genetics of people controlled versus people who have progressive disease. This requires large numbers of genetic material from lots of different people, and here the goal is to compare genetic material from 1,000 HIV controllers and 2,000 people with progressive disease. And what you do is you look for small genetic markers that are enriched in people who control that aren't present in people with progressive disease. The main analysis is looking at HIV controllers, both viremic and elite controllers, versus progressors. And then there are secondary analyses looking at, for example, just elite controllers versus chronic progressors, and long-term non-progressors versus chronic progressors. And after at least the preliminary analysis of that data showed that there in fact is a region within the genome that is associated with control, and that all occurs within this area called the MHC. And right smack dab in the middle of that is HLA-B57. So HLA-B57 is something that has been known to be associated with control. That's been known even before these genomic studies were done. But what's interesting is that, and that result, confirmed results, had been shown in another similar study that was put out by David Goldstein and team. And what's kind of interesting is that, at least with this preliminary analysis, well, first of all, MHC seems to be important. MHC helps determine how what parts of the virus your immune system attacks. And so it suggests that really there are very important things about the immune system within controllers that determine control, that there's something genetically going on there with the immune system. And that, right now at least, there doesn't seem to be a lot else. Although with more numbers, with more patients enrolled, with more genetic material, you have more sensitivity. So the hope is that as the numbers increase, there may be other hits that we see that give us other clues. So part of what's being done now is that the enrollment numbers are going up, but also an attempt is being made to sort of merge the data from David Goldstein and other groups who've looked at these sort of genome-wide association scans to try to get more sensitivity. Okay, so, and I just want to, I put this slide in to remind myself to say that it's not just the virus, and it's clearly not just the immune system, it's clearly both. So when I said that elite controllers had less fit virus, so if you looked at those controllers and those progressors who actually were B57 positive and pulled them out, what you see is, in fact, even amongst controllers or progressors, people who had B57 had less fit virus. So clearly, that less fit virus is probably also, in part at least, a function of the immune pressure being created by the immune response, so that controllers may be able to drive the virus so that it's less fit. It's not simply that they get infected with a less fit virus and that's sort of the fortuitous chance that happens. No, there's clearly a role of the immune response in creating that less fit virus. Okay, and I just want to touch briefly on and change gears a little bit and talk about HIV in the gut. And Barbara's going to talk a lot about this, so I won't go too much in depth to this. But as Peter touched upon, there's been a relatively new appreciation that HIV has a lot of effects on your immune system in the gut. And everything we know about the immune system the immune response against HIV is based upon studying immune cells in the blood. But in fact, most of your immune cells are in the gut, the lining of the gut. So anywhere from 60 to 70 to 80% of your T cells are actually in your gut. But it's a hard area to study because it means getting biopsies, right? And as Peter also mentioned, one of the consequences of HIV infection in the gut is that you have this massive depletion of CD4 T cells in the gut, and you have this leakiness in the gut, which then leads to sort of bacterial translocation and may have a role in more immune activation. So one of the things that we've been involved in is looking at some other consequences of that leaky gut. And we know that HIV controllers have lower levels of expression of this. I have 1 minute left, so I'll try to wrap it up. But they have lower levels of expression of this inhibitory receptor called KLRG1. And people with progressive disease have higher levels of expression of that. Now, what activates KLRG1 is a molecule called E-cadherin. And people who have HIV clearly have more E-cadherin in their blood. But where does E-cadherin come from? Well, one of the places it comes from is from the gut. And what you see when you stain for E-cadherin in the gut is that normally E-cadherin forms, its role is to form the glue between the layers of the lining of your gut cells, and they help prevent leakiness in the gut. But when you have HIV infection, that gets destroyed partly, and you sort of get this redistribution and solubilization. So that gets broken up, and then E-cadherin gets released in the gut, gets released in the blood, and it's detectable in the blood, and it's higher in people with HIV infection. Because we know that people with progressive disease also have higher levels of the receptor that binds to E-cadherin, what we see is that one of the consequences of that higher level of E-cadherin is that you have more dysfunction of your T cells caused by, presumably, E-cadherin binding to KLRG1, this inhibitory receptor on T cells in your blood. The consequence is that those T cells are less able to secrete cytokines, and they're less able to kill infected target cells. So it leads to a model in which, other than one of the other consequences of leaky gut due to HIV is that you have this solubilization of this molecule E-cadherin that binds to inhibitory receptors that stops your T cells from doing their job, which is fighting infection. And that inhibitory receptor KLRG1 is expressed at a higher level in people with progressive disease than controlled disease. And then the last slide, I just want to touch upon one of the real barriers to better understanding what's going on in the gut is the fact that with these biopsies you get very few cells. And the question is, how can you really maximize the amount of information you get from those very few cells you obtain from these gut biopsies? So we've been working with this group at MIT, headed by Chris Love, to try to develop new technologies that I think are really exciting and really interesting. What you can do is you can have a little grid that's about the size of a stick of gum, and on that grid are about 200,000 individual little wells. And in each of those wells, you can take immune cells that you have from, for example, a tissue biopsy. And then you can simultaneously ask lots of different questions of those cells. You can, if you look down here in the lower left, it's kind of hard to see, but these are pictures of cells. Sorry, these are on the part labeled D. These are pictures of cells in these little tiny microscopic wells, and they're labeled with different fluorescent markers to distinguish what type of cell they are on the surface. So we can tell if a cell is a T cell or a B cell or a macrophage. We can also simultaneously tell what cytokine that individual cell is producing in response to particular HIV stimuli. And we can also tell whether or not that cell is capable, if it's a CD8 T cell, capable of killing an infected target cell. And it doesn't show up there on the right, but that's sort of the general concept of these new technologies. So that's it. Basically, the gist of it is that, you know, the gut is, there's a lot of things going on with HIV in the gut, and it's an area of great interest. Barbara, you know, is clearly going to talk more about that. But I think that there's a real need for new technologies to try to maximize the amount of information we can get from studying that area. I just want to thank all the people at the Ragon Institute and our collaborators, and of course, all of you for participating and helping making these studies possible. So thank you.
Thank you so much, Dr. Kwon. So interesting.